Hello, hello, hello. I'm back, and this is lecture number 15 from Human Biology. It's called Transcription and Translation. It's kind of a twofer. And these are two steps that we're going to talk about that take us from DNA to proteins, because I've been saying over and over again that the DNA carries the instructions for making proteins. This is about how that happens. We know how it gets replicated. We know how it's built. We need to try to figure out how to make those enzymes from it. And it involves another molecule called RNA, which I've introduced to you. And there's some funnies here. I think they require some explanation though. So let's start on the left. I don't know if you guys are Game of Thrones fans, but this is from Game of Thrones. This is Daenerys Targaryen. And she is one crazy freak, but that doesn't have anything to do with this. What this is it's a joke that involves her hair. Over here, let's see if I can get a color that shows up. Okay, over here we have RNA, which is a single helix, a smaller molecule than DNA. And look, her hair strand here is one single twisty turn. And over here on the right, her hair is a double-stranded twisty turn, and that's double-stranded, double helix DNA. So that's really all the joke, and it says, I will take what is mine and with replication and transcription. I don't know if she even had a line like that, but one crazy freak. Okay, the next one also shows, wait a minute, I need more color. It also shows double-stranded DNA and single-stranded RNA, and they're in the nucleus, and this is an indication that the RNA, the single-stranded RNA, is a messenger for the DNA. In fact, we're going to meet some messenger RNA, and this RNA has a little messenger bag, and he's pissed at his boss, who seems to be smiling, and apparently his name is Arthur. I don't know why. Arthur, the mRNA, was at the end of his strand at work. mRNA is messenger RNA, so we're going to meet that, and this is supposed to be funny. It's even like in a circle because it's the nucleus of the cell. Oh, that's funny. And then the last one is another cat science meme, and the cat is saying, if I had a choice between DNA and RNA, I'd choose RNA because it has U in it, and U is uracil. And I think I mentioned before that RNA doesn't have the same nucleotides as DNA. DNA has A and G and C and T. RNA has A and G and C and U. Uracil instead of thymine. So that's why that's a funny joke. Got it? Let's move on. All right, so we have seen this slide before, but when we saw it before, I said it was a preview. So here it is for real. Let's remind ourselves that we're showing what is called the central dogma of molecular genetics here. It is a major central idea that explains how the DNA carries the instructions for all of those proteins which control all of the functions of the cell. So we have two processes here and three different molecules that we need to deal with. The first is DNA. We know a lot about that. That carries the instructions. Somehow that gets changed into RNA, and that happens through a process called transcription, which is kind of like transcribing from one language to another. You might hear that, or maybe you've heard of medical transcription, where the gibberish that the doctor talks about is kind of written in English for the rest of us by people who can understand what the doctor is saying. Anyway, so transcription is taking the information in DNA and putting it into another form, a form that is a smaller molecule. We saw that RNA was a single-stranded molecule, smaller, and it can get outside of the nucleus. So we go from DNA to RNA. Those are both nucleic acids. They are both polymers that are made of individual nucleotides, okay? So once we're outside of the nucleus, the RNA is going to find itself on a structure called a ribosome, which we saw when we did the eukaryotic organelles. And on that ribosome, the RNA, which is called messenger RNA because it's carrying that information, is going to be read, just like during transcription, the DNA was read, but it's going to be read, and it is going to be made into not a polymer of nucleotides, but a polymer of amino acids. And when amino acids are strung together into a polymer, that makes a protein. So we've got translation happening on a ribosome outside the nucleus. 3 different macromolecules, 2 different procedures, but we start with DNA and we make our way to proteins. It involves RNA and DNA, so it bears mentioning again how they're different from each other. The difference molecularly is that the sugar is different. The sugar in the molecule for RNA is just ribose, whereas the sugar in DNA is deoxyribose. What's the difference between them? Well, you might notice that here there's an OH and here there's an H. So there's an O missing because it's deoxidized, deoxyribose. It's really not that complicated. There are, remember, 4 different nucleotides that make up DNA, A, T, C, and G. They are a little bit different for RNA because instead of T, it has U. It has uracil. That's what that science cat was saying. I like RNA because it has U in it. Okay. So let's start with transcription. Right away, you might notice it looks an awful lot like DNA replication, because with DNA replication, the 2 strands of DNA split, they opened up, and then this enzyme came in that was called DNA polymerase, and it looked kind of like a big orange blob, and it came in and it read the DNA and it brought in nucleotides to make another strand of DNA right next to the template strand. Well, in this case, the big orange blob is RNA polymerase. It's a slightly different enzyme, and in this case, instead of grabbing nucleotides to build another DNA strand, it's going to grab nucleotides to build an RNA strand. But it's only going to do it on one of the original DNA strands instead of doing both of them at the same time. So that's what we're looking at here. The DNA is opened up, and the RNA polymerase has come into position, and it is reading the DNA and bringing one nucleotide at a time into position so that it's creating a strand starting at this end. And so this is the place where it's already made some of the strand. It's getting longer and longer at this end, okay? And that's what we have up here in this cartoon kind of looking thing. I thought it showed a little bit better how the DNA opens up and the RNA polymerase, which in this case is 2 green blobs working together. They are reading the DNA, moving from left to right and leaving a single strand of RNA behind them as they read forward through the DNA. So again, it looks a lot like DNA replication. That's what it says up here. Very similar to DNA replication. But it only transcribes the gene regions of the DNA. It turns out that there's a lot of DNA that doesn't have any genes in it at all. Sometimes they call it junk DNA. Sometimes they call it nonsense DNA. It turns out to be very useful when we're trying to do some scientific things, because you don't want to be studying DNA to identify things if that DNA is involved in some type of genetic process. So we can use the junk DNA in order to solve crimes with the FBI. We can use it in order to figure out who the daddy is. We can use it in order to figure out how closely related 2 species are. It's the non-genetic DNA that we use for that. The only other thing on here is this idea that we start from a promoter region, which is a special spot on the DNA just before the gene. So it's kind of like there are a bunch of nucleotides that say, if you're reading in this direction, you're about to hit the gene. So the RNA polymerase knows that that's where it starts to build the RNA. And in this case, as I said before, it is messenger RNA. And so that's that Arthur guy who is being made to go out of the nucleus and take the message of the information with him. All right, this looks good. Transcription. All right, so I've got a little thing here that I need to explain that forces me to take a step back, because I've been talking about transcription in the nucleus, translation outside the nucleus, but these things both happen in bacteria, and bacteria has no nucleus. So I want to mention how this happens in prokaryotic cells. In prokaryotes, which have no nucleus, what happens is the RNA polymerase jumps onto the DNA and starts to do the making of the single-stranded RNA. And then what happens is there are ribosomes that are just floating around in the cell. So these red dots in this cartoon are ribosomes. And what it's showing is that transcription and translation are both happening in the same place. As soon as this RNA gets made, the ribosomes jump on it and start to make polypeptides, which we're not showing here. We're just showing the blue strands are the RNA and the red dots are the ribosomes, and they would be making little strands of polypeptides, which are strings of amino acids. And it would look like some crazy hairy mess, which is what we have over here with an electron microscope picture of transcription and translation happening inside of a prokaryotic cell. What we have, let me see, I can use the colors. Here's green, here's the green DNA in the middle, just like in my little picture here, okay? And then there are blue RNA strands coming off of it, okay, just like that. And there are little red, wait a minute, I gotta make this thing smaller. There are little red ribosomes attached. That doesn't look red to me, okay. Or maybe I got purple on there. Here's red, wait. Woo, I'm doing art just for you guys. There's the red ribosomes, okay. And coming off of those, which we don't have a color for, is a whole bunch of little strands of polypeptides, proteins being made. It's all happening at once. So it is a crazy, hairy biochemical mess. No nucleus, all happening in the same place. I'm sorry that I jumped right to the eukaryotic cells. That's where I'm going now. But please keep in mind that it's all happening here in the prokaryotes. Okay, so here we are back in the eukaryotic cell where things are happening in 2 steps. And that's what I'm going to explain now. So over here we have the nucleus, and all the DNA is inside the nucleus, and outside we have the rough endoplasmic reticulum, which is all covered in those little red ribosomes. And so we're going to have transcription happening inside the nucleus, making RNA, messenger RNA, and then that messenger RNA is going to leave the nucleus through these little tiny pores. And the first thing it's going to run into is the ER with ribosomes all over it. And so that's where it's going to meet the ribosomes. 2 different places for these processes to happen. But before we can get out of the nucleus, there's a couple of extra steps in eukaryotes that doesn't happen in prokaryotes. So let me introduce those. After we've made the RNA, it has to be processed. It's processed. It's cut up and put back together and arranged just right, so that when it meets the ribosome, it's all prettied up, okay? So what happens? First of all, there are sequences that are called introns, and if I were going to write this science, the ones that were called introns, they would be the ones that I would keep because they're in, right? But in this case, the introns get tossed. And then there's another group of pieces of RNA called exons, and they get kept. I don't know why you would toss the introns and keep the exons. Maybe it's because the introns are staying in the nucleus. They're getting redigested and the exons are getting out. I don't know. But anyway, the introns are removed. The exons are left together and spliced back together. And that's what we're showing here. It's called RNA processing. All we have left is exons. Those are actually the genes that are going to be used. So the introns are some more of that junk DNA that gets made into junk RNA that then has to be removed. Okay, so we remove the introns, splice the exons back together, and then we do a couple of weird things on one end, and it is the 3' end. We put what's called a poly-A tail, where it says adenine, 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 adenine. Those are all in RNA, and that tells the ribosome where the end of this chain is. And then at the other end, we have a very strange nucleotide called methylguanosine, which isn't in the list of our 4. It's a very special one because it marks the other 5' end. So we're cutting out the introns, putting the exons back together, and putting some special things on the ends to get this all processed. If there's any accidents or mistakes that happen with that, we're not going to make the right protein. So it's a very important process. It's almost like proofreading and getting it ready again. Okay? And then finally, the last thing on the list here says that the primary transcript leaves the nucleus and recruits to the ribosome on the endoplasmic reticulum. Let's say it's that ribosome right there. I just picked that one out. I like that one. Boom. That's where we're going to do translation. So transcription happens in the nucleus. We make the messenger RNA. Messenger RNA leaves the nucleus, finds a ribosome. Now we're going to do translation. Okay, so here we are outside of the nucleus and getting involved in this translation. And right away, we see that in addition to the messenger RNA, we have two other forms of RNA. So we say that there's lots of RNA involved. Messenger RNA is carrying the genetic information, and then the ribosome itself is actually made of RNA. We call that ribosomal RNA. And then there's a third form of RNA called transfer RNA, which is a little tiny a twisted molecule which is carrying around an amino acid on it. In this case, here is one of the, let me get a different color. Here is one of the messenger RNA molecules, and it's carrying around this amino acid on it on a string like a balloon. And this amino acid is tryptophan, which is one of the 20 amino acids that we can be carrying around. But transfer RNA always carrying that around, and even if it's dumped off its load, it's going to go and get another one. So it's used over and over again. All right, so we've got the messenger RNA, the ribosomal RNA, which is actually in two parts. The ribosome's in two parts. We say that there are large and small parts to it. They're called the L and S subunits. L for large, S for small. They come together, and we can see that down here. This is the L, and this is the S. And the messenger RNA strand is kind of sandwiched between them. They start reading at the 5' end. Okay, they start reading right here, and then this whole mess is going to travel in this direction like a ticker tape being read. I don't know if you even know what a ticker tape is. It's like a strand of paper that's got something on it that you're reading as it's going past you, okay? And what it's reading is the nucleotides that are in that RNA strand. A, U, G, U, G, G, U, U, C. You notice it has U's instead of T's, okay? So that's the RNA. Each amino acid corresponds to a 3 nucleotide sequence. So the transfer RNA, that's what we have over here, okay, which is carrying amino acid, that's tryptophan right here, has something on this end which is called an anticodon. An anticodon. It's a set of 3 nucleotides or 3 receptor sites that correspond to 3 nucleotides. And so it recognizes the nucleotides on the RNA and bonds with them. You'll notice that U bonds with A bonds with U, and C bonds with G right there. So it bonds with them, and when it bonds with them, it lets go of its amino acid. And that amino acid gets tagged onto the end of a chain that's being made. And then the transfer RNA molecule is released, and it floats off to grab another amino acid somewhere. So am I missing something here? Each amino acid corresponds to a 3 nucleotide sequence. The genetic code. So tryptophan, okay, that's this one over here, is always ACC, which is what we see there. Okay, that's actually the anticodon. I think what I'm looking for is the codon, so it would be UGG would be tryptophan. We'll see that when I go to the next slide, whether I'm right or not. Anyway, what you need to know is that each amino acid has certain 3 nucleotide sequences. Those are called triplets or codons. And all life, which all involves DNA, all life has the same set of codons that apply to these 20 amino acids. So the next slide I'm going to show you is the 20 amino acids and all the codons that go with them. And it is called the genetic code. It is the genetic code of life. It's not just a metaphor. This is what all life is based on. Bum, 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 the drama. Okay, here it is, the genetic code. This is for all living things. There are 64 possible triplets with the nucleotides that we have, and there are 20 possible amino acids. So the numbers don't come out perfectly, the 20 amino acids and the 64 triplets. So often the third nucleotide is a little redundant, which means there are a bunch of codons that apply to that all apply to the same amino acid. And the one example that I've talked about is tryptophan right here. And tryptophan is right here on the genetic code. And as I said before, the codon for it, the triplet for it, is UGG. The anticodon, which is right here on the transfer RNA, is ACC. And so that would be looking for the complementary pairs, which are right here, UGG. So it's looking to move into that space right there. And by the way, on the ribosome, there are 3 spaces. Maybe I'm getting a little ahead of myself, but it's showing them there. There are 3 spaces. There's the A, the P, and the E space. That is read APE. Even though it says EPA, it's APE because A is the first space that the transfer RNA comes into. P is the second space that it moves into, and then it releases its amino acid. And then E is the last space it goes into and exits. So I think of them as the access point, A, the polypeptide point, because we're making bonds, and then the exit point, APE, so that we can see that it's moving across there. Again, tryptophan here is UGG, but there's lots of other amino acids on here. This is phenylalanine, this is leucine. Leucine has a lot of codons. Look, these are all for leucine, but you'll notice most of them start with CU and then have the third one as being different, but also these UUs over here are also leucine. Here is methionine. That's a very important one because that's the one that all the polypeptides start with. Look, here's methionine right here. It's the first one on the chain. So what's going to happen is this transfer RNA molecule is going to move in here, and then it's going to attach tryptophan right underneath this methionine so that then they're part of a chain, and that chain is going to grow just like the RNA grew when we were transcribing it from the DNA. Okay. What else do we have here that you might know? How about lysine? Lysine is a common amino acid that you might have in your nutritional supplements. This is alanine. This is valine. I don't know if you've heard of any of these. This is serine. This is arginine, and there's a bunch of arginines up here too. Anyway, this is the genetic code, okay? 20 amino acids, and for every combination of 3 letters, there is an amino acid that goes with it. That's why I have that game on the wall in the lab where you throw those little Velcro balls at the dartboard, and there's 3 of those balls, and so if you get 3 of them in a row, it's kind of a study guide for trying to figure out what is the amino acid that goes with that 3-letter set, that codon, that triplet, the genetic code. Okay, so this is the 3rd slide in a row that I've had this picture on it, and it's there because I like it. It really shows the pattern, and there's just a couple more things I want to say about translation. The small subunit holds on to 3 codons at a time. This is the small subunit down here. And then we have this, I've said all these things already. We have the binding sites here. This is called the anticodon on the messenger. I'm sorry, on the transfer RNA. See, it says tRNA, which then is going to bind right here with the codon on the messenger RNA, and as a result, we're gonna go into the A, and then tie the polypeptide together at the P, and then exit the ribosome at the E, and the whole mesobob is going to be moving in this direction. So we're moving toward the 3' end of the, then that's the poly A tail, remember, the 3' end of the messenger RNA. So again, I'm kind of restating it, showing here again one of my favorite pictures. Okay, so this is a complicated process, and when we are trying to describe complicated processes in science, we make confusing diagrams. And I have some examples here of those confusing diagrams. Remember that we're starting inside the nucleus, and that's where transcription is happening, where DNA is being transcribed into RNA. That's this green strand, which then leaves the nucleus and finds a ribosome, and the ribosome parts come together, and the transfer RNA is coming over and adding one little amino acid at a time, which is making a long strand of those amino acids. It's making a polypeptide. Polypeptide is what a string of amino acids is, before it folds up into the functional protein that it's going to be. All right, we knew that because we knew that the monomer for proteins was the amino acids. But if it's just a string, it's called a polypeptide. When it folds up, it's called a protein. The next example we have here is over here where we're inside the nucleus again, doing transcription. We're making this strand of messenger RNA. It goes out through the nuclear pore, and then it's finding a ribosome. And in this case, instead of the little purple jelly beans there, we've got a whole bunch of like gumballs of different colors, and that's because there are 20 different types of amino acids, so they're going to show them with different colors. And it's making a growing chain, which is a polypeptide, and then it's going to fold up into a protein. All right, just representing the same process so that we can get a great kind of sense of it. This last one I don't really understand, and it makes it even more confusing because they call it a newly born protein. I don't know where that's coming from, but this is, I think, supposed to be a more accurate depiction of the shape of a ribosome. And these blue things here, these weird blue like monster looking things, are transfer RNA molecules. And you can see that each one of them is carrying an amino acid, and they're coming in. Look, the A is on this side this time. A here, and then P here, and then E here. And what's going on? This is where A is recruiting it, and then P is making the bond to the previous amino acid, and then E is where transfer RNA exits. And as a result of doing that, you look, this is the ticker tape stuff that's all moving in this direction as it's being read. Anyway, I know that's kind of creative, but I don't know that it adds much to this. But my favorite diagram of this whole process, which isn't going to make it any clearer until I label it, is this next one. Oh my goodness, what is this mess abob? It's crazy. I feel like I'm in space. This is some kind of weird science fiction thing here. It's like creatures on another planet. No, it's not. It is the process of transcription and translation happening inside a cell. And the reason I know that is because I was able to label these things. So where do we start? We start with DNA. That's down in here. Oh, my purple isn't working very well. And then that gets transcribed into RNA. The RNA is heading out through these nuclear pores. That, look, here it is. Here, look at the RNA coming out of the nuclear pores. And then it finds a ribosome. Maybe I can use a pen instead of a highlighter. Here we go. Whoops, that's not right. Yellow, yes. There we go. It finds a ribosome. Translation happens on the ribosome. We are reading in this direction. And these are the transfer RNA molecules that are coming in, and they're carrying these little purple balls on them, which are the amino acids. And so the amino acids are being linked together into a long polypeptide, and then it folds up back and forth and up and down in helices into a protein. We even have a mitochondrion in here. That just makes me so happy. Anyway, I don't know whether this helps or not, but I thought it was a great piece of artwork. Okay, well, if after that you're feeling like I just can't take any more of these strange, confusing diagrams with all the blobs and chains and different shaped, colorful things, that's okay, because I'm just about done. I just wanted to add a couple more details to this. And it might clarify exactly how that endoplasmic reticulum is involved and where the ribosomes come from. It turns out that the ribosomes are actually floating around in the cell, and when they bump into some RNA, they are able to travel along protein strands to go to special pores on the endoplasmic reticulum. All the pictures of the rough endoplasmic reticulum we're showing, they show ribosomes all over them, but there are ribosomes floating around outside it also. The reason that's important is because when the ribosome finds that pore and attaches, as it makes the polypeptide, okay, it's going to make it and dump it into the endoplasmic reticulum, to the other side of the membrane. So the ribosome stays on the outside, the polypeptide gets dumped to the inside, and it folds up inside the endoplasmic reticulum, and then this whole mess is going to form a little vacuole and travel off somewhere to the Golgi apparatus or one of the other places in the cell where those proteins might be required. And that's why the endoplasmic reticulum is so flippy-floppy and folded over so much, because it's constantly spitting off these blobs which contain the polypeptides that were created by the ribosomes. That's pretty cool. So I think I made this clear, and I've got one more picture that I thought you might find interesting. I thought you might like this picture because, you know, we did that kinesthetic thing with the giant pool noodles doing the chromosomes, and I wanted you to know that I've been doing this for a while here at Columbia College. And this is another group of my biology students who did a kinesthetic thing, but in this case it was transcription translation that we were modeling. And what we did was we used 3 chairs as a ribosome, and each student had to carry a balloon. They were the transfer RNA molecules. So they had to carry a balloon, which was their amino acid, and they had to sit in the chairs and then twist the balloons together in a chain to make a polypeptide. And then this is 3 polypeptides twisted around each other in a triple helical form, which is collagen, which is a very important protein. So these are some of my biology students who made a collagen molecule by doing a kinesthetic version of transcription translation. This took a long time, though. I don't think we could do that. And by the way, this person over here, this is Jessica Brooks. She's one of our librarians now. That's how long I've been here. Oh my goodness, that makes me feel old. Anyway, hope you're doing great this week, and I'm done. And be safe, and I'll see you in class.